I want to move on with the show, so I'm going to take the next slide, which shows you our agenda today. The welcome is what I'm actually have just done, a little bit of housekeeping, and then I'm very pleased that we have Professor Joseph New. He's the coordinating investigator of the Connection study, Professor of Pediatrics at the University of Florida in Gainesville. He will start the show, and then after that, I will join in and give you latest news out of the Connection study, the information that we can share with you, and we give you hopefully a lot of information which you might not have.
Thank you, Eamon, and it's a real pleasure to be here this morning and to be able to talk to you about this. What I'm really going to focus on is necrotizing enterocolitis and a little bit about feeding intolerance and how the Connection study might relate to these problems. So could I have the first slide, please? The agenda is that I'm going to talk about necrotizing enterocolitis versus imposters. Even now in 2020, we still don't have a really good definition for necrotizing enterocolitis. And this is one of our major problems in terms of moving forward with this disease. We have various pathophysiologic pathways that we think are related to necrotizing enterocolitis. And one of them is dysbiosis, and I'll talk about dysbiosis a little bit more later. And I'll also talk about neonatologists possibly causing dysbiosis and actually causing some problems in the neonatal gastrointestinal tract and the types of microbes that are there. And then we'll talk about feeding tolerance and then the introduction to the Connection trial. Next slide, please. So here at the top of this slide, we have a picture of a baby who's born at around 27, 28 weeks gestation, and the baby is on mechanical ventilatory support, and that baby comes off of mechanical ventilatory support, and about 3-4 weeks later, the baby develops this very distended abdomen. This is a typical picture of necrotizing enterocolitis. And once this happens, it's usually a surgical situation, and the risk of death is 20 to 30%. And so this is a very, very significant problem that we have in neonatal intensive care. As neonatal intensive care has evolved over the last 50 or so years, we have not made much progress in this disease. And one of the probable reasons is that we're also saving smaller and smaller babies, and the risk for necrotizing enterocolitis seems to be higher in the smallest of babies. But there's also other problems that we have. One is, as I mentioned before, we lump several diseases that we call necrotizing enterocolitis into the same data set. So the data sets that we have to retrospectively evaluate necrotizing enterocolitis are dirty data sets. And so there's many different diseases that we are actually looking at and that we're calling necrotizing enterocolitis. We have animal models that really don't represent the disease that we see in human preterms. And one of these animal models is the rat asphyxia model, which I actually used during my fellowship back in the late 1970s. And there are now about 250 papers in PubMed using this particular model. And when we really look at it carefully, this model does not represent necrotizing enterocolitis. We've also tried to focus on several different particular pathways, such as the TLR4 pathway, that leads to necrotizing enterocolitis. And certainly there's a strong chance that that particular pathway is involved, but this is multifactorial. And there are several different pathways that lead to this disease. And so we are now developing new bioinformatic approaches where we're able to actually do systems biology and look at the whole picture to see how certain diseases develop. And this is one of the things that we're also doing with necrotizing enterocolitis. Next slide, please. So here are some of the NEC imposters, and I put at the top stage 1, 2, and 3 necrotizing enterocolitis. These are the Bell's criteria that were developed in the late 1970s in Kansas by Martin Bell, who was a pediatric surgeon. And I think that all of us in neonatology are now recognizing that stage 1 necrotizing enterocolitis is such strange situation that so many babies with just feeding intolerance are diagnosed as having necrotizing enterocolitis. Stage 3, we have babies who develop pneumoperitoneum who don't have true necrosis of the intestine. They have spontaneous intestinal perforation, and those babies in the past have been put into the same bucket as necrotizing enterocolitis. So as you go down this list, these are various imposters that we have for necrotizing enterocolitis. So I think that we're beginning to recognize more and more that the data sets that we have for necrotizing enterocolitis really represent more than one disease. Next slide, please. So here is a picture of what I would call classic necrotizing enterocolitis. And we have a baby at the top who has a distended abdomen with some periumbilical erythema. And X-ray was taken, and if you look at the arrows on the radiograph, you see the bottom arrow represents pneumatosis intestinalis. It's pointing to pneumatosis intestinalis. At the top, you're seeing portal venous gas, and this is a baby who continued to deteriorate, was taken to surgery, and you can see at surgery the baby had a necrotic piece of intestine that was taken out, and that baby actually did fairly well. But most of these babies who need surgery for necrotizing enterocolitis end up having significant problems, not just in terms of short bowel, but neurodevelopmental delays, and there are many other problems associated with the development of this disease down the road after these babies survive for longer periods of time. Next slide, please. So we're also beginning to recognize that there's a mean gestational age at which necrotizing enterocolitis seems to be at its highest. So at the top picture here, you see a 23-week gestation preterm baby, and at the bottom you see a 29-week gestation preterm baby. And so the question is, when is this baby on the top, the 23-weeker, most likely going to develop necrotizing enterocolitis compared to the 29-week baby? And you can see this graph, which is a graph that I looked at or developed with Mohan Pami at Baylor University. And you can see that there's a peak that occurs between around 28 to around 30 weeks corrected gestational age. And so what this means is that the 23-week preterm baby will develop necrotizing enterocolitis usually 6 weeks down the road, whereas the 29-week preterm baby may develop at around 2 weeks down the road. Next slide, please. So what are some of these hypotheses that this generates? Well, there could be a change in the microvasculature. This is a developmental issue that may be occurring. So for example, another disease that we see in preterm babies, retinopathy of prematurity, usually doesn't occur until a certain gestational age, usually around 30 weeks corrected gestational age. And so the microvasculature might be involved with this. Toll-like receptor developmental pattern. We know that the last part of the second trimester of pregnancy, if you look in the gastrointestinal tract, toll-like receptors are at their peak. And then we have microbiota changes that also occur. And I want to focus a little bit more on the microbiota changes. Next slide, please. So here is a picture that I want to spend a little bit of time on. Back in around a decade ago, we started to get a lot of interest in looking at the types of microbes that are present in the gastrointestinal tract using newly developed sequencing technologies. And these newly developed sequencing technologies, the price of them started to come down, and so we were able to determine the names of the microbes in the gastrointestinal tract prior to the development of a disease such as necrotizing enterocolitis. So for example, at the University of Florida, Mark Hudak, Renu Sharma and I, we started some studies on evaluating the microbes in preterm babies prior to the development of necrotizing enterocolitis. And several other groups did the same thing. Erica Claude at University of Chicago, Barbara Warner at Washington University. Several other groups started to look at the same thing. And so several papers from individual places, universities were published, and then Mohan Pami from Baylor Medical Center contacted me and asked me if we could possibly put all these together into a meta-analysis. So take the sequence data and put them all together from all of these different centers. And this is what we did. And the slide that we see here is the result of looking at several different centers, 16S sequencing prior to the development of necrotizing enterocolitis. So on the figure that you see on the top, on the left, you see the control babies, and on the right, you see necrotizing enterocolitis babies. And you see a change in the colors, and these colors represent phyla of bacteria. If you look over time in the control babies, you don't see that much change. But then when you look at the necrotizing enterocolitis, you see that the blue begins to increase and the green decreases. The blue represents a certain phylum of bacteria called the proteobacteria, and the green represents the firmicutes. The proteobacteria are largely your gram-negative microbes, and these are the bacteria with a lot of LPS, lipopolysaccharide, in their cell walls. And those are the ones that increase over a period of time in those babies who develop necrotizing enterocolitis. These are the toll-like receptor agonists, the LPS in the cell wall of these bugs stimulates toll-like receptors. The green represents the firmicutes. Firmicutes are largely gram-positive microbes, and the gram-positive microbes are microbes that produce short-chain fatty acids, such as butyrate, acetate, propionate. And for example, butyrate is a very important short-chain fatty acids that maintains tight junction integrity between epithelial cells. We also see this small difference in the yellow. If you look at the control babies, you see at the very bottom some yellow components of these bar graphs. You see almost nothing in the babies with necrotizing enterocolitis. And this represents a phylum of bacteria called the Bacteroidetes. And you can see at the bottom, the bar graph, there's a significant difference between the necrotizing enterocolitis Bacteroidetes versus the controls. The Bacteroidetes are interesting because in their cell wall, they contain a protein that actually stimulates the production of certain kinds of T cells that are tolerizing T cells. And so in the control babies, you see more of this capability to produce these tolerizing T cells than in the necrotizing enterocolitis babies. So what this means is that there seems to be this association between a dysbiosis, different kinds of microbes that develop in babies who develop necrotizing enterocolitis versus control babies. Oops, next slide, please. So are there some practices that we have in neonatal intensive care that actually may lead to the development of necrotizing enterocolitis? And here we see some of the causes of inappropriate colonization that lead to dysbiosis. A lot of our preterm babies are not fed enterally for fairly long periods of time. That has been associated with the development of a dysbiosis in the gastrointestinal tract. The type of diet, using formulas rather than human milk, or even donor milk rather than the baby's own mother's milk, may contribute to a dysbiosis. Antibiotics also are very important. Next slide, please. And as we know, antibiotics are the most commonly used drugs in the neonatal intensive care unit. And this is a slide from the pediatrics group looking at their data set. Ampicillin and gentamicin are by far the most commonly used antibiotics in neonatal intensive care. And as we know, most preterm babies in their first couple days get at least usually about 2 days or more of ampicillin and gentamicin or penicillin and gentamicin. So that, we think, leads to a dysbiosis in the immature gastrointestinal tract. And there have been some observational retrospective studies that have linked the length of time on antibiotics to greater risk of necrotizing enterocolitis. Next slide, please. The other area that we are looking at in the connection study, in addition to necrotizing enterocolitis, is feeding tolerance. And feeding tolerance is very important, and this is our primary goal, along with necrotizing enterocolitis in the feeding study. And we know that if we cannot enterally feed babies for a prolonged period of time, we have to nourish them by using parenteral nutrition, and that is associated with complications such as cholestasis, infection, sepsis, increased risk of BPD, increased pulmonary vascular resistance. Also, lengthening the time of TPN increases the cost of hospitalization. Next slide, please. Over the past several years, we've undergone this safety and tolerability trial, and I think that several of the people on the line here have actually been involved with the safety and tolerability trial. And one of the things that we have seen here is that the adverse and severe adverse effect profile was similar in the active and in the placebo groups. And there were no severe adverse events related to the study drug. Also, there was no evidence of cross-contamination with the study drug in comparison to placebo. And the key conclusion was that it was safe and well tolerated. And now we have the go-ahead from the FDA to go into, next slide, please, the phase 3 trial. And here's just some of the data from the phase 2 safety and tolerability trial. We had different cohorts, and I'm not going to go into all the detail here, but again, there were no significant differences in terms of adverse events between control and the study drug. Next slide, please. Here are some other adverse events that were looked at, and again, no differences in the different cohorts in the safety and tolerability trial. Next slide, please. So now we are getting to the phase 3 of the connection study, and this is going to be one of the few prospective randomized controlled studies done in neonatal intensive care using a probiotic-like agent or drug. We consider that this is a well-designed study. It took a long time to put this together. It's got good scientific value, and the 2 primary endpoints are necrotizing enterocolitis and time to sustained feeding tolerance. Next slide, please. We know that this connection study will help us because there are so many uncontrolled studies out there with uncontrolled probiotics, and these have been messy, and the meta-analyses of these are actually quite messy. And so to do this in a prospective, randomized way is, I think, the right way to do it, and hopefully we can together move this field forward. Thank you for your attention.
Thank you, Joe, for that great presentation. Now let's look at the connection study a little bit more in detail. What's happening? Where are we? And what's the latest news? So first of all, before we start, I'd like to say I hope that everybody online, everybody's families, and indeed all the people at the NICUs where you're all working, and indeed at the hospitals, are in good shape, and that you're weathering this particular storm that the COVID-19 virus is wreaking havoc across the world. So that's number one. And then the second thing, I just want to reiterate a little bit Joe talked about, the background, the medical background of the whole thing. But I mean, we at IBT, we are now testing in the, and rather you, in the connection study, we're testing the candidate drug product IBP 9414. Now, this has been developed by IBT probably over the last 7, maybe even 8 years now, when I think back the time that's going, but certainly 7 years. And we've been developing it all the way in order to make sure it's the safest possible, let's say, drug candidate that we could possibly give to these premature babies. We know that they are extremely vulnerable, and we are doing absolutely everything we can to make sure that this product is as safe as it can be going into these babies. And I say that because it is in contrast to all the food supplements that you hear. And the food supplements can have very many different shapes and forms. They can come as additives to mother's milk, and they can come as additives maybe within mother's milk. None of them that are out there are developed in the way that we are done, with the safety in mind, to the level of safety that we've developed this drug, potential drug. And how we do that is we do it under IND in the US, and we do it with the EMA, the European Medicines Agency, and under CTX, let's say the equivalent of IND in the European countries and in Israel. And they're developed under the scrutiny of those medicines agencies. And the things they look at are the things that are bulleted here. You can see CFR 21, CFR 210, 211, guidelines for how you should get dose accuracy with a single dose vial that we have, stringent control of bioburden and microbial purity of the product, according to Pharmacopoeia, both in the US and Europe. All this is a lot of language which you don't use every day, but which is something you all expect of anything you want to give to a premature baby in a very vulnerable situation. So, having said that, we are now at phase 3, and we're in the connection study. So this is the first ever Regulatory Medicines Agency approved phase 3 study. This is the only one of any live bacterial therapeutic in premature infants. So we're unique in that sense. As you know, we have regulatory approval to go into phase 3 in the countries that are represented online today: Hungary, France, Spain, Israel, UK, and the USA. And to date, there is no live bacterial therapeutic product that has been approved by a Regulatory Medicines Agency for use in premature infants. Now, what I want to do is give you some numbers. Here's a map of some parts of the world. We have today 82 selected NICUs as part of the connection study, 54 in the US, as you can see, and then we have 9 in Hungary, 6 in Spain, 5 in France, 5 in Israel, and 3 in the UK currently. The next slide is going to show you the geography. Here you can see 2 different colors. So the ones that are green here are sites that are open and recruiting babies currently. And the ones that are orange are ones that we are preparing to open. So they're very close to opening, but they're not quite open yet. And really, that's maybe one of the little issues that we have. We need to get these orange stars converted to green stars as quickly as we possibly can, because we need everybody up and running and recruiting babies into the trial to make the trial a success, so we can show what we need to show, or try to show. I would point out here, Hungary is doing very well. You can see Hungary with a lot of green and recruiting sites, and we congratulate them. They've been very, you have been very supportive of the trial and doing very well in the trial. Hopefully we'll get the remaining 2 sites open quite soon, and we'll have everybody on board there. And then we have a great deal of sites, a large number of sites in the US. And here, and again, when I show you this slide, I apologize to anybody who's been geographically dislocated. Either you may be in the wrong part of the state, or you might be out in the water. I'm not sure, but I tried to get everybody on board here. And again, you can see a lot of green sites now that are actively recruiting babies and going as quickly as we can to get this trial going and moving. We also have a lot of orange stars in a lot of places that we'd like to get converted over. But you can see a concentration of sites up in the northeast, on the eastern seaboard, all the way down to Florida, which is probably our best site, or most compacted site when it comes to NICUs that are involved. A lot in the south, a lot in Texas, a lot of coming on board in Texas. up in the Chicago area, in the Midwest, and then over on Los Angeles, mainly in Los Angeles on the West Coast. So let me go to the trial. How is the trial going? So now, if we look at the progression of the trial, you will recognize maybe these two arrows as the beginning part in the protocol, the first two categories of babies that we staged the recruitment of the babies. So the first part, and many of you have talked about this en route and you wonder about this, the first part of the trial recruits only babies between 750 and 1,000 grams at birth. That's by design, and we've designed this with the authorities and everybody. So this is by design, and we can't change that, so to say. That's the way it has to be to make the trial work. And what we want to do is get to the DMC, and the DMC will, the Data Monitoring Committee will convene when data from 300 babies is available. And as you can see, we're kind of approaching halfway there. So we're getting we're well on our way to getting to 300, but we're not at 300 yet. So we still have some way to go. And really what we need to do, I mean, we got a lot of momentum, I have to say, in the January, February period. And then really the momentum really January. And then in February and towards March, things slowed up a bit. Maybe due to COVID, probably is very much due to COVID. But still, we have a lot of things to do. And I think what I would say, in order to try and get us all to 300 as quickly as we can, so we can open up this larger population of babies, we can open up for the 500 to 1,000 gram birth weight. Those of you who are recruiting babies and have had a first patient at your NICU, thank you very much for the work you're doing. You are recruiting at very good rates. And then thirdly, we need to get as I said before, the orange stars need to get to be green stars. So we need to get everybody who's not quite started yet, started as best we can and as quickly as we can. If we do all that and we push all this along and we get through COVID, or we do this parallel to COVID, then we can get this 750 to 1,000 arrow filled up to 300, and then we can go through the DMC and hopefully move on to a bigger population, which will allow for more babies to come in quicker. So really, anything we can do there and that's my little slogan, which comes here, which really, to make connection work, if this study is going to work, we really need every baby you can get. On the clinical operations side, things are working well. I mean, study procedures seem to be working well. We have screening, consenting. The dosing within 48 hours doesn't seem to be a problem across the board. Study drug preparation administration seems to be going as we expected and according to plan. We've had dialogue with quite a few sites about that, but no major problems, and things are going well. And the AE and SAE, the adverse event collection process, is working as we expected and satisfactorily, and we appreciate that. So that's kind of a message for us. If we can use all these processes to get recruitment moving, then we should be in good shape. A few words on safety. So far on the safety side, there have been, in the study, we've seen 6 deaths. None of the deaths have been considered to be related to study treatment. So that's good and expected. There have been no related SAEs reported to date related to the study drug. And safety observations are in line with expectations of what you would expect, both from the phase 2 trial that we did earlier and also from the 750 to 1,000 gram birth weight population that we're now looking at. So things seem to be as you would expect in the population. So we're not seeing anything peculiar there in that sense. Thank you very much, everybody.